Thank you. Next session is tracking COVID-19 with a system clinical approach. With that, I'd like to pass to Lee Hood, the session chair. Currently, Lee is the Senior Vice President and Chief Strategy Officer for the Institute for Systems Biology and a Senior Vice President and Chief Science Officer for Providence St. Joseph Health.
I'd like to welcome you to a session on tracking COVID-19 with a systems clinical approach. And certainly the previous session with Joel Dudley gave us much to think about from a computational point of view. And in this session, we'll be exploring various new kinds of technologies. We have three outstanding speakers, and they will go in order: Lars Steinmetz, Ida Grundberg, and Kevin Hrusovsky. And I think you'll see some both fascinating results from technology and COVID, and maybe even some hints about how COVID has changed our future and how we think about technology. So with that, Lars, would you like to begin?
Yes, I'm happy to start. So I'm here representing two hats. In a way, I co-founded SOPHiA GENETICS in 2011, and I'm also professor of genetics at Stanford University and senior scientist at EMBL. And I'd like to tell you a bit about what we've done and how COVID-19 has impacted our work, both at the company, but also in my own labs. SOPHiA GENETICS is a global health tech company that was, I said, founded in 2011 and has really worked on integrating healthcare institutions across the world with now 1,000 healthcare institutions and diagnostic labs across 85 countries. And in total has analyzed over 600,000 genomic profiles from patients, with about one patient every 4 minutes coming online. Initially, it was launched to be a multi-omic platform technology, but genomic data was the first that really entered the system and has since expanded to radiomics data and clinical trial data, now also electronic health record data. And in future, the company is interested in expanding in proteomics, which is also fitting given our representation on this panel. And altogether, it uses advanced artificial intelligence and machine learning algorithms to do pattern recognition across data in the system and also to develop prognostic predictive models. The whole platform and the data is available online, so a clinician can log in and see this information. This also enables insights gained from a patient from one part of the world, say in the US, to benefit from analysis carried out on a patient with the same disease in a different country, because annotation is shared across the system. And so that enables better patient analysis and also patient management. Initially, most of the data in the system has been, and actually still is, from cancers. Somatic and inherited cancers make up about 80% of the data in the system. 20% come from other inherited diseases, including rare diseases or metabolic diseases. But of course, when COVID-19 hit, we saw an opportunity to utilize some of our technologies to benefit the study of COVID-19 and to provide better management for these patients. And in two ways: one, to develop algorithms for advanced analytics of COVID-19 viral genome sequences for better variant detection, and also to use the radiomic technologies to distinguish patients with more severe or more mild forms of COVID-19. I'll show you one data slide for each of these. So for the whole genome sequencing pipeline, SOPHiA GENETICS developed an amplicon-based sequencing approach for COVID-19 and launched this in a study across 6 sites of clinical partners with which it had already been working, and developed metrics, quality control matrices, to assess how much read depth is necessary to detect a variance in these genomes. And with about 1,000 reads per sample, or 1,000 copies of viral genome per sample, one can get over 89% coverage of the genome and detect alleles down to 10%. And that gives you information about where these variants arise. You can use that to track transmission of COVID-19 and also see where new mutations have arisen. So for example, the spike protein, we've heard about new variants coming from England and also South Africa and Nigeria that show variants in these positions. And of course, even though we have vaccines, continuing to monitor the genomic sequence of the virus will continue to be important to make sure that these vaccines continue to work, but also to identify other therapeutic or diagnostic approaches. In addition, we've used the radiomic capabilities of SOPHIA, which include using machine learning algorithms across image data. In this case, it's CT image data from lungs of COVID-19 patients, which are then segmented and then analyzed in order to both distinguish COVID-19 lung samples from other types of lung disease samples using machine learning methods, but also more interestingly, I think, to distinguish between very severe COVID-19 patients from less severe ones, based on CT image data that was collected on the first visit of these patients to the hospital. So the algorithm was able to distinguish from these image data patients that eventually would enter the ICU or die of the disease versus ones that have more mild forms of the disease where they could leave the hospital or even could go home, or just standard of care types of analyses. In addition to the work at SOPHIA, my lab has also been working on novel ways to diagnose this disease using next generation sequence technology. And in my lab at EMBO, we've developed a multiplex PCR amplification approach to diagnose COVID-19 that we've launched to monitor our workforce as the scientists are coming back to the lab. This approach uses extracted viral RNA that's then amplified through a multiplex PCR with unique molecular identifiers and then ultimately sequenced. And with this approach, we could distinguish positive COVID-19 clinical samples from employees of EMBO that have gotten tested, all of which are mostly negative. And we get a good distinction here. One thing that we did to drive the cost down was to home purify all the enzymes that are needed to carry out this reaction. That allows us to do a library prep per sample of 92 cents. And depending on how many samples you sequence and multiplex, you can get costs of 1 to 5 euros per sample, but you can multiplex thousands in a single reaction. In another implementation and in a collaboration with Li Hood, and Sui Wang at the Institute for Systems Biology, we've been working on developing a home test for COVID-19 that uses a LAMP reaction, a linear isothermal amplification of the viral genome, and then, which we think is unique here, an impedance-based detection of the viral sequences through sequence-specific hybridization events that happen within this electrical detection chamber. The whole device is small, portable, and should cost somewhere between $1 to $2 to manufacture. And this is, of course, early stage, and we're developing that data now. And so that's all I wanted to talk about in the presentation. I'd be happy to discuss any of these data and to come back to any of it. Thanks.
Well, thank you very much, Lars. I think what we'll do is go through each of the 3 presentations, and then we can have some general questions and so forth. So if we could turn to Ida, our second speaker now, you're on.
Thanks again, Lee, for this opportunity to talk about our work with broad proteomics profiling in COVID-19 patients. So first, just a very brief recap that we still, almost a year into the pandemic, don't fully understand the underlying cellular mechanism causing the immune response and the outcomes of these infections. And we also lack a comprehensive overview of the inflammation processes, but we know for sure that there are significant variances in disease severity, but there are still many unknowns why some people are suffering from more severe outcomes than others. Also, this is a disease that is impacting multiple organs and causing both short and long-term effect. But we have been very fortunate to have been very close and intense collaboration with the scientific community. So we realized very early in the pandemic that what was needed was deep profiling of the immune system to be able to find predictors of severity and also find ways to stratify patients based on outcomes. We also need to find novel therapeutic targets as well as re-evaluate existing ones. So we can use that to treat patients and also find how the impact of these therapeutics are on organs. So we as a proteomics provider, we have from the beginning been very committed to support these studies. So our library covers secreted proteins, organ-specific markers. We have been very fortunate to collaborate with Lee and his group at ISB. We have a very broad library of inflammatory proteins with very high sensitivity of the low abundant markers that are especially key for these type of studies. And we also have a number of established drug targets and candidates from ongoing trials in our library. So our platform is based on the proximity extension assay. So what's unique with that is that is based on dual antibody recognition together with DNA barcoding. So this provides very high specificity. Then we have amplification to get sufficient sensitivity, and we have digital barcode using either NGS or qPCR. Also, what is quite unique is that we're using very limited amounts of samples, so just a microliter of patient material. And now with using NGS, we have very high throughput, so we can screen thousands of samples per week. So our library today covers 1,500 proteins, but in the coming years, we are reaching the 3 and 4 and 5 and so on 1,000 proteins. It's fully validated, so we have been very careful about that and have also built in a quality control system. Also, it's scalable, so our studies today are used for discovery, but we're also working more with clinical implementation. I'm happy to discuss more of that later. And since the beginning in March, we have run now hundreds of COVID studies. And I wanted to bring up one example here. So in the spring of last year, we teamed up with the COVID-19 Technology Access Framework and partnered with the great teams at the Mass General Hospital and the Broad Institute, led by Dr. Marsha Goldberg and Neera Cohen. So in this study, we screened 1,500 proteins in around 400 patients. And as part of this initiative, all data was made public. So since September of last year, everyone can access about 1 million protein data on this site. So the cohort consisted of plasma samples from 306 COVID positive patients and 78 COVID or symptomatic COVID-19 controls. And samples were taken at baseline, which was the time at the emergency room, and then day 3 and day 7 for the COVID positive. And then the WHO's classification was used based on outcomes within 28 days. And you can see the groups here. So there was the patient that died, patients getting intubated, those that were hospitalized with oxygen supply, those without oxygen, and those that were discharged. So the first thing we did was to compare the profiles of the infections, so the COVID infected ones with the symptomatic controls. And we found more than 600 differentially expressed proteins associated with the infection. You can see that here in the volcano plot. So what we saw that the COVID positive patient, they displayed higher expression of proteins associated with viral response and interferon pathway proteins, and also associated with vaccine response, innate immune activation, and T cell function. So you can see many interferons here. We also, maybe interesting, we also saw decreased levels of CCL16 and 24. You can also see a heat map here with the top 200 proteins. We then, moving on, we continued looking at severity associated proteins. So here we compared patients from group 1 and 2. So those that were, they were defined as severe, and group 3, 4, and 5 as mild. So first of all, here you can see in the heat map, they significantly change proteins between severe and non-severe at day 0. So in total, we identified more than 250 proteins that were separating these groups. And we can look more closely into some proteins, as you can see here, that changed over time, over the course of the collection time, and also how the association to overall survival, like IL-6 and IL-8, CCL2 and IL-24. And then finally, perhaps the most optimal result would be a predictive model for severity or outcome. So we built a classifier based on our protein data. So the result was an area under the curve of 0.85, which is considered as good performance. And then also we looked into plasma signatures, all pathways mediating ARDS, so acute respiratory distress. We could also use the signature to stratify the most severe, so those that died from severely ill patients. And you can see the top significant proteins here. I think there were 24. But yeah, I don't have time now, but there are a number of other examples too. But I think to summarize, we are very, very encouraged by this data and see that we can actually use plasma protein profiles to be strongly predictive of disease severity. And it really highlights the potential use for both in clinical trials to enrich for patients, but also to tailor therapy to specific interventions. But I think I will stop there and then leave for more discussions later.
Well, thank you very much, Ida. So Kevin, would you like to tell us about how we can use highly sensitive protein analyses to analyze COVID?
Absolutely. Thank you very much for this opportunity. Let me see if I can get the slide up. Are you able to see the slide?
Yes. Yes.
Awesome. Very interesting discussion from Lars and Ida, and we really believe very much in the protein and the role of it. And certainly, Lee, over the years, you've been the pioneer that has taught a lot of us and trailblazed in this area. Our company, Simoa technology called Quanterix, is basically going after exquisite levels of sensitivity in measuring the proteins. And by doing so, using digital technologies, we are able to, we think, transform the way health is administered by seeing very early on disease, long before symptoms, and also to do it non-invasively. And so this is a transformative area, and independent of Quanterix, we have an ecosystem called Powering Precision Health, where all the world leaders in oncology, neurology, and now infectious disease, including the major agencies, NIH, CDC, FDA, et cetera, working diligently to try to advance the field of proteomics. Historically, we've spent a lot of time in neurology by seeing brain health with a very simple blood test. And that was where we really started to evolve the technology. Now there's thousands of third-party peer-reviewed publications, of which at least 300 or 400 of them are focused just on brain health, being able to see it in venous draws, but also dry blood spots. And most of the companies shown on this slide are utilizing it to recruit patients very early in the disease cascade, long before symptoms, for their drug trial cohorts. And particularly in Alzheimer's, where we've now been able to see it 16 years before dementia, it's playing a major role in getting to precognitive impairment cohorts that are highly specific for Alzheimer's, by being able to rule out Lewy bodies dementia, as well as frontal temporal dementia, utilizing multiple protein markers in a panel. So now that is really the way we had been focused when several outsiders came to us saying, what can you do for COVID given this crisis? And so we quickly pivoted our company towards COVID. And we now have a fairly high definition roadmap. But once again, what we learned is that there's a lot of false negatives and false positives in today's COVID testing. And this is particularly true if you try to measure COVID before symptoms. So once again, deploying the same technology that we had been using for brain health and for oncology to see COVID before symptoms has led to many breakthroughs over the last several months. I'm going to click this slide and it'll just show you, when you are able to see it earlier on this curve, where you can see the early stage of the actual cytokines first peaking. And then you see the antigen starting to elevate in the blood. And then ultimately you see the adaptive immune system respond. And we now have the ability to measure across all of these various components and these various assays and their peaks. And that has led to some pretty significant disruption over the last several months. So it started with us being able to measure the cytokine storm and predict who was going to go into the storm by seeing the interferon alpha very early. And now with our sensitivity, we're actually looking at 8 different subtypes of the interferon alpha. So there's been many publications in the last couple months just talking about the ability to stratify patients around the world in the hot zones. We ran a webinar on precision health through the hot zones. Many of the scientists showed how by seeing interferon alpha too low or too high created the storm. And so being in that middle was something that panel helped the field see. Then we started to see loss of taste and smell that led to sometimes brain fog. And so our NFL, neurofilament light, is able to measure that. And there's now several neurological presentations and publications on the loss of taste and smell and being able to measure that with our NFL and moving that now to long haulers, trying to understand the long-term impacts. Is this going to create early stage Alzheimer's, Parkinson's, MS because of the neuronal death? And so the NIH, as well as the FDA, have been working very closely with this. The NIH has given us $20 million over the last several months to scale our tests up. They were so very significantly promoting what we were able to see in blood of the virus, that really led to some pretty big breakthroughs. We did get our EUA about 3 weeks ago for the serology tests that we can run. Here you can see we can quantitate semi-quantitatively the IgG, but more importantly, our founder, David Walt, who's using this down in Brigham Women's, the same technology, we can actually run a 3x4 looking at 3 different IgG, IgM, and IgA across 4 different viral components. So this has been an amazing way to kind of get a better picture of that adaptive immune response. And then the most important area is the antigen itself, where we're the only ones that have been able to showcase it in blood and see it in blood almost at times when you can't even see it in the respiratory. And lately in the long hauler campaign, there's been a lot of speculation that for those that can't get rid of their symptoms over a 6-month period, that the virus is going dormant in the stomach at low levels, and it's no longer in the respiratory. So they're testing negative, but the symptoms keep returning. So now there's a lot of work out in the NIH. Anthony Fauci's group under Cliff Lane now has our technology, and they're running around the clock looking at it in blood. So seeing the virus move from one part of the body, respiratory, into the gut, and then being able to see that small level of the virus, and it comes back, the adaptive immune system crushes it for a while, and then the virus starts to grow back again, is the speculation. And so a lot of those types of studies are now being researched. And so having a comprehensive look at this virus across the innate immune system, the adaptive immune system, as well as the antigen and the serology componentry, we think has been really helpful, and we're pretty excited about continuing that advance. And the whole area of proteomics, we think, is just beginning, and Quanterix sits kind of at the end of the pipeline before drug trials. We're trying to advance further into the diagnostic sector, but ultimately we do love a lot of the diagnostics and the discovery of proteins, which we think today it's only 5 to 10% covered. And so the things that we heard from Ida and others, we think are incredibly productive for the future of this field. Thank you very much, Lee.
Oh, thank you, Kevin. So I'd like to address to each of you just a general question. You've told us a little bit about the kinds of things that you can do with your technologies. My question relates to a more general question of, has the social and financial imperatives of COVID really pushed you in new and different directions, directions that you can see could be applied broadly to other things in the future? And, just a brief answer is all we're looking for. So Lars, do you want to start?
Yeah, maybe I could say, so from Sophia's perspective, I mean, one of the things that certainly had helped us, I think, respond to the pandemic and, keep our genetic diagnostics operational was the fact that a lot of it happens in the cloud and is based on compute servers that, are maintained centrally, but not locally, at a more global level. So this didn't require shutdown to happen as it did, in my academic labs where, things really went, stalled for some time until we were allowed to go back into lab and restart these projects. So I think one of the things that we've learned from COVID-19 is to deal with that challenge of not being able to operate locally, but move more of our systems into a cloud-based setting and to operate there. Now in Sophia, that was already the case in an academic lab setting. We had to move to a lot of things, with remote calls and focus on manuscript writing and grant preparations before we could move to new data collection. So certainly that has had an impact on both sides.
So let me ask one more question, Lars, and that is with the emergence of the British mutant, there is a real imperative for sequencing viral genomes in everybody.
Yeah.
Do you think we have the technologies to manage that with what you described, or do you think we have to go beyond what you've done even?
Well, I think certainly there is room for more improvement, right? I would always say that, being an interested individual in technology development and focusing my academic labs in that space. Though I think we have products already that are there to do large scale COVID sequencing, and there are collectively over 300,000 COVID genomes in public databases already. So I think a lot of that data, though, still is riddled with some false positives and errors. And it would be probably good to redo these with more accurate sequencing approaches. And that's why kind of Sophia put a focus on benchmarking the accuracy of these whole genome sequencing methods across different sites to come up with recommendations, including kind of how deep to sequence and what you'll be able to detect. Because I think one challenge is certainly low variant allele fractions. We know that this virus mutates. There's a new mutation arising every 2 weeks. And possibly even in individual patients, as the virus propagates, new mutations will arise, giving rise to heterogeneity in the sequence. And being able to detect all that really does require, approaches that focus on quality control and understand the limits of detection in the resulting data.
Thank you. So Ida, how about your thoughts?
Definitely, it has definitely have an impact on us as well as at Olink in many different aspects. So first of all, we have 1,500 proteins, but we have realized that we need to go beyond that, which was the plan already before COVID-19, but now we're trying to speed up the process even more. So that's why reaching 3,000 this year and trying as fast as possible to reach the whole plasma proteome. But also we are trying to see, since our technology is very flexible and scalable, so we are now on 2 different alternative platforms, so we can be more agnostic to have the outreach of our technology on more sites using both next generation sequencing and qPCR, also in different formats and ways of running broad proteomics, so high plex to low plex and relative and absolute quantification and more custom based. So we're trying to listen to all the feedback that we have received. Also what Kevin was mentioning, more home sampling, that's of course is very interesting area as well, to be more prepared for the next time. So can we do more to help maybe more outpatients and not having patients going into the ER, but maybe predict that before and can tell who's going to stay at home. So definitely it had an impact on us, and we think that it's exciting to be part of this and trying to, again, be prepared for next.
So a question for you then is, you have focused on the plasma proteome, and I agree, that's absolutely the proteome you want to start with. But what about the other proteomes, things that don't necessarily get into the blood? I mean, do you have plans for being able to look at those proteins as well?
So we do definitely, plasma and serum are the main sample matrices that we are running, and also what we have focused on and done all the validation on. But we do also a lot of work on CSF, for example, like what Kevin presented, but also other matrices. I think what I was also mentioning, the small volume requirement that we have of a microliter, that opens up a lot of opportunities in pediatrics and in small tissue biopsies from tumors and so on. So we're also doing more and more work and optimizations on other matrices. And again, with home sampling, filter papers, dry blood spots, different devices where you collect minute amount of samples. So the technology is very flexible for basically all types of sample types, but our focus has been blood.
So how about single cell analysis? That's another really critical area, obviously.
Yeah. So we have done some studies. There are some papers also on single cell proteomics with our panels. So that works. It hasn't been our focus, but it's definitely an interesting area that we, I think, at some point will move into as well. But they are both collection where you can source or sort single cells and then run a panel. And a microliter is very small, so as long as you have that, we can run it, and the sensitivity is quite impressive as well. So yeah, it's definitely doable.
So Kevin, you gave us a lot of directions that COVID is pushing you. A, I wonder if you had any further comments, and B, I'd be interested in how broad the spectrum of proteins are that you can analyze, because one dimension of opportunity is sensitivity, but another opportunity is being able to do global, that is systems-driven analyses. So I'd love A, your thoughts on any other directions you've been pushed, and B, in a sense, how do you see scaling up so that you can look at more than the limited number of proteins you're visualizing now?
You've asked some profound questions, and we are, first of all, to your first question around socialization, we had this vision that if you could see proteins with exquisite sensitivity, there's a lot of ways you can translate sensitivity into productivity, even multiplexing. You give up a lot of sensitivity when you multiplex. And so getting to the more system category, we think continuing to persevere and drive, and we just did another 100x on top of the 1000x, we think we can translate a lot of that sensitivity into some profound understanding of the multiplex, which does give you the systems approach. And I got to tell you, the Zoom era is what probably transformed us the most, because this precision health ecosystem around low invasive testing to see disease before symptoms has just exploded, with people being able to link in with us from around the world on every topic that we've had. And COVID has brought it to the forefront. We're a publicly traded company, and our valuations have gone off the charts, even though we really haven't had tailwinds from COVID yet. This is really even around the Alzheimer's and around the things we're doing inside the body. Many of the same companies, the Lillys of the world, are then shifting over into COVID with a lot of their synthetic antibody treatments, or the Gileads looking at the remdesivir. And so across the board, people are looking at these proteins. And you made a comment that I thought was profound, and that is, are you going to be able to look for proteins that aren't in blood? And I could tell you, when we first started, a lot of people said, you're not going to find the proteins we're finding in blood, because they stay inside the cerebral spinal fluid. There's the blood-brain barrier, that there was a belief that it doesn't cross it at times. And we were able to find these proteins at very low concentrations and correlate them to what's in the cerebral spinal fluid. So that then opened up home care sampling, which transforms neuro health, because now you're able to do with home care sampling, dry blood spots or even saliva, to see these proteins at exquisitely low sensitive levels. So the large payer groups now have discovered us, and they're the ones that the United Health groups and the folks that have 70 million members, they're interested to kind of find disease very early so they can economically treat it and then extend life health. If you can extend life expectancy by getting the disease before symptoms and treating it, you can transform healthcare. So to me, the payer groups are really found us through the Zoom over the last 4 or 5 months. And now I think we're onto a path where the protein is going to become a revolutionary opportunity to transform healthcare, which was what we've all known, but COVID has brought it to the forefront in my mind, Lee.
Yeah. So let me just push you. How many different proteins can you detect? And what are your plans for expanding your repertoire of detection?
So right now we have this thing called Homebrew, where we allow our customers to devise their own ability to measure it in our system. And so we've got probably 80 to 100 proteins that are off the shelf. Now we've got, diagnostic for, SARS. We've got the antigen. We just got the EUA actually 2 days ago for the antigen test, and we got the serology. Those are the only two that are diagnostic, but we probably got 100 that are off the shelf kits. And then many of those are multiplexed into maybe 6-plex. But then our customers have found another 200 or 300 that they've used our technology for. So we're only sitting today at probably 200, 300 proteins. But I actually believe what Ida's up to is like the next generation of opportunity. We saw Sears go public. I'm the chairman of another company, 908 Devices, which is a little handheld mass spectrometer. So I think the whole field of proteomics is only 5 to 10% covered today. And if you can get to the low invasive testing in home, prevent all the infection carrying of having to get exposure, I think that you could get to thousands of proteins. And I think Ida's already at 1,500. Sear suggests that they might be at 5,000, or maybe SomaLogic feels that way. I think that there's at least 10 to 15,000 that you should be able to get to. And when you do, I think you're going to have big translational opportunities that dwarf what's been going on in genomics. And our founder founded Illumina, so he actually is very compelled by the protein. And so I think it's the future. And I think you had this vision way at the beginning, Lee, and I think it's starting to come to fruition right now in 2021.
Yeah, it's an exciting time. I agree. So, Lars, let me ask you about the home device you mentioned. What do you see are the real advantages in that device vis-a-vis having everyone have in their home a simple detection device for nucleic acids and things like that?
Right. One of the advantages of the design that we've currently drafted is that since it's based on sequence detection, it would be purposable for other pathogens as well. You can design, probes that detect a variety of different sequences. And so with the same kind of detection technology, you could potentially do more than COVID-19. That's one of the attractive areas. The other way you could multiplex is do multiple patient samples in parallel through, parallel impedance chambers, and therefore scale kind of the assay dramatically, potentially to several thousands. And the fact that device, as we've currently designed it, is entirely portable, and you could either make that on a single device or on two devices, where you could have the impedance readout sensor on one device, and then a cartridge that actually contains the chambers that are exposed to the sample, which could be disposed. You could, make the actual test on a day-to-day basis, even, in the cents range, once you've purchased, a single detection cartridge. And we think that home testing is certainly, as it was brought up, from both Ida and Kevin, home analysis is going to be important because there's so many asymptomatic carriers out there. And if we can detect, the virus earlier and can detect patients that carry the virus earlier, then even on top of the immunization that occurs, this will provide an additional control, because I think it's going to take some time until the vaccination, the immune, population-based immune response is going to be broad enough in order to curtail further transmission of the virus. So I think these things, we need both, and I think we'll continue to need home-based testing. I think Lee just dropped off. But maybe I'll ask you a question, Kevin, that kind of came up in your presentation, and Ida touched on it too, in terms of protein detection and the ability really to detect the disease as early as possible. And I fully agree with you. The more sensitive we can be on protein detection, the better. is it going to be possible to drive this, to be as early of a detection technology as would be with nucleic acid-based detection of the viral genome? That's currently the gold standard. And have you done some analyses there to compare the two?
We have, in fact, that was actually the initial seminal publications that came out, was that this wasn't the ability to see the protein at the same sensitivity as DNA, which was really one of the game-changing components. And it's been real interesting in looking at the number of copies of the virus and COVID, and there are standards down at the FDA that we're working with Tim Stenzel on that deal with, seeing it with, quantitative PCR, and then what we can see with the antigen. And, we're pretty much at equivalent levels today. And we think that that's been one of the game-changing components to what brought DNA to where it's gotten to. So being able to do that repeatedly and in a democratized way by making instruments that can be democratized out throughout the world, repeatedly get to that sensitivity with the protein, is what we think is creating the rapid trajectory of value creation in the proteomics field. So I actually think that you're absolutely right. And we've got some of the publications that show the ability to even get to digital PCR levels of sensitivity with the Simoa as a protein analytics, and then quantitating the protein. That's the other big piece of this, is the quantitation for like these IgG markers and antigen markers. We're seeing huge stratification opportunities just in being able to measure how much of the antigen. We can almost predict how severe someone's going to be based on the level of antibodies opposite the amount of antigen. So they're looking at those indexes of those two to be able to make those kind of predictions. And quantitation, I think, will be key to helping unravel much of the virus's mystery right now.
So I apologize. I'm on Friday Harbor with a low bandwidth internet, and occasionally I drop out and everything. But I have one more question for Ida, and that is, there is one other company that has large-scale protein analysis, SomaLogic, and I'd love to hear your comparative analysis of the two platforms.
Yes, so they have an excellent technology, or an excellent solution to antibodies that, of course, can be limited, and we are, of course, limited to antibodies in our case, and we are using 2 for each assay. So using aptamers could be a great solution to overcome that. We have had kind of different strategies. We have since the beginning been very focused on the technical performance and doing a lot of validation for each of our assays. So it has been taking us longer time to reach the thousands of assays. But of course, there are already comparison and the correlation between the two platforms out there. So there are published papers as well. So there are some overlaps, and there are some assays that are not overlapping, and also some good. I think what is also a strength that we haven't had time to talk about yet here is also the work that is going on in the proteogenomics space. So we are doing more and more of that. So you can also see how protein expressions are linked to genes, and that's a good way to also do orthogonal validation of 2 comparative proteomic platforms. Yeah. So I encourage the audience to look at some of those correlations. But again, I think our focus has been on validation and assuring high quality from doing a lot of testing on technical performance. It takes a bit longer, but we believe in quality.
Okay. Do any of you have questions for one another? I have one more comment that I would make, and that is, I think one of the enormous, just barely emerging frontiers of biology is single cell analysis. And it seems that really all the technologies we've talked about here have unique possibilities for contribution. But again, questions of the scale of proteins or nucleic acids that can be measured and their accuracy, and most of all, the cost and so forth are really critical kind of questions. And I'm just wondering if each of you sees this as an opportunity, or I'll ask a more general question. Do you see other opportunities we haven't really thought about deeply emerging from COVID, where we ought to think about the analysis and integration of information from different kinds of informational molecules or so. Very broad, very general question, but I'd be curious what you think. So, Lars?
I think maybe I'll keep it very short, but mention two things that came to mind. One is, which I already touched on earlier, is the ability to, using nucleic acids to multiplex and detect multiple pathogens in parallel. I think if we had the focus that we've placed now on COVID-19, worldwide, not only us, but everybody, in developing detection technologies, if we had done that 20 years ago, I think we'd be much more, rapid and more quick off the blocks here for COVID-19. And the same types of technologies that we're developing for COVID-19 can be repurposed for other pathogens. So I think there's a tremendous opportunity going forward in detecting multiple pathogens, including, organ-specific biomarkers in blood through, analysis of proteins, for example, that we've heard about too. I think there are tremendous opportunities within that space. And the second area is in vaccination. I think the mRNA-based vaccination, also again, a nucleic acid-based technology, because it's sequence-based, can be repurposed to target other types of diseases, and it's been applied for cancers and certainly for other pathogens, I think, is an opportunity too. So I see two things that really, have entered into the big arena from COVID-19, but provide a lot of opportunity in the future.
Thank you. Ida, any comments on your side?
I can only speak from the proteomics side. So we have tried to piggyback on the genomics trend with the cost revolution they have done, and that was one of the reasons why we also decided to go with NGS as a readout. So that will, we think, at least enable our community to run larger studies. And then just the second one that I also think is great to see how COVID has had an impact on the society, that we can do more, share more data and do it more in open access and publicly available to speed up the process and not sit on data, but actually share and help each other out. So that's.
So Kevin, briefly, since I think we're almost at the end of our time.
I do think heterogeneity is key in getting to the single cell, and then being able to measure how many proteins are on each of those cells is critical for the future. I really am a huge proponent of disease prevention, and I think that the protein offers so much opportunity for us to all empower our own ability. Someday having biomarker watches that are real Fitbits on steroids, I think, is the future to navigate your own health journey with proteins in the future. So great session. Thank you.
Well, I want to thank each of our speakers. You've brought us distinct visions of the opportunities COVID has presented. And you, I think, have all clearly expressed where COVID is going to be pushing us in the future and how much of what we've done is going to be applicable to other diseases. And I think one of the most interesting comments made today by Kevin was the idea that Zoom has given the ability for collaborative interactions at a scale that were utterly inconceivable prior to this time. So with that, I'd like to thank everybody, and I hope you've enjoyed a superb panel.